The cell membrane is a low dielectric medium because it is essentially consisting of hydrophobic acyl chains of lipids. Now, if you go back to basic electrical circuits, you will remember that a capacitor is essentially an insulator sandwiched between two conducting plates. If this capacitor is connected to a battery, electrons will flow in the circuit, and this flow of electrons will continue until the capacitor is fully charged. And during this process, charge builds up on the two sides of the capacitor. Now, if you take a cell containing 0.1 molar potassium ions, and the outside concentration of potassium ions is also 0.1 molar, then no ions are flowing across this cell. If I immerse this cell into a medium containing a lower concentration of potassium ions will start flowing out through the cells through these channels, which are called resting potassium channels, which are always open. Note that the concentration of potassium ions will not be the same on both sides of the cell at equilibrium, because although the chemical gradient pushes potassium outside, the negative electrical gradient of the cell inside with respect to the outside opposes the outward flow of potassium ions. Therefore, an equilibrium will be established. Under these conditions, there will be positive ions accumulated outside the cell membrane, and there will be negative ions accumulated inside the cell membrane. And this looks very similar to the capacitor in this circuit. And now the membrane is a capacitor which holds charge. We will return to this equilibrium between the chemical and the electrical gradients in a moment. So after potassium ions have diffused out of the cell through these resting potentials, through these resting potassium channels, then an equilibrium membrane potential is established across the cell membrane. The cell membrane is then thought to be called polarized because it has a electrical gradient across it. And this electrical gradient is mainly established by potassium ions, and this is not correct. Leak potassium channels generate this membrane potential. So there are two forces here. One is the chemical gradient. So if this is the membrane, the chemical gradient points in this direction, while the electrical gradient points in the opposite direction. And one can now make a quick calculation of this electrical potential, the electrical potential that will be established under these specific concentration gradient. So at equilibrium, there are two free energy sources. One is the work done by moving 1 mole of ions across the cell membrane, and this is given by the electrical potential multiplied by Faraday's constant. And this must be equal to the free energy, or the difference in the free energy of the concentration differences across the cell membrane. So this is the free energy, this is the concentration of potassium outside divided by the concentration of potassium inside. If, of course, your ion is not a single charge but has two charges, then this would be divided by nu, where nu would be the charge on the ion. We can also call this Em star because that represents an equilibrium resting membrane potential. If you make this calculation, if you just plug in the numbers, you will find that the equilibrium membrane potential, resting potential, resulting from a difference in potassium ion concentrations across the cell is approximately equal to minus 87 millivolts. The typical value of the resting potential in cell membranes is about minus 70 millivolts. There are other factors which contribute to this membrane potential because, for example, there are many negatively charged proteins inside the cell which are not present outside the cell, and so on. There's a gradient of sodium and chloride ions as well. But it is mainly the movement of potassium ions and the concentration differences of the potassium ions that generates this resting membrane potential. This resting membrane potential then is also equal to what is called the equilibrium potential of potassium, also called in some cases the Nernst potential of the potassium ion, because we are in this case looking only at potassium ion concentration gradients. Okay, so now we have established that at equilibrium there is a resting membrane potential across cells, and this makes the inside of the cells negative with respect to the outside. In order to generate these membrane potentials, another protein that must be involved is the sodium potassium pump, which pumps out 3 sodium ions and pumps in 2 potassium ions for each molecule of ATP hydrolyzed. The sodium potassium pump also compensates for osmosis effects. So if you look at the cell in image A, the concentration of solutes is higher inside than outside. If this is the case, then water will flow into the cell through water channels called aquaporins, and the cell will start to swell and eventually it will burst. However, the sodium potassium pump pumps out 3 cations and pumps in 2 cations, resulting in a negative electrical potential inside. And because of this negative electrical potential inside, negatively charged chloride ions flow outside the cell, thus balancing the osmotic pressure. So you have an equal concentration of solutes outside and inside the cell. And this image here at the bottom shows you very important data, that the concentration of potassium is low outside the cell, high inside the cell. For sodium it is the opposite, and for chloride also it is the opposite. So now the cellular membrane has electrochemical gradients across itself. So there's the chemical gradient of ions across the membrane, as well as an electrical gradient established by potassium leak channels and the sodium potassium pump. Okay, with this background knowledge we can move on to neurons. So neurons are essentially cells which convey information in the form of electrical signals. So if you look at the yellow neuron here, it has various parts. These are called dendrites. Dendrites are responsible for receiving signals from another neuron. This part is called the central cell body, and this cell body is responsible for processing these electrical signals received by the dendrites. Below the cell body is what is called an axon hillock. You don't have to remember these names, just understand the concepts. This is where this electrical signal is generated. And the function of the axon, this yellow tube here, is to transport this electrical signal along the neuron. You also have axonal branches right here. Which lead to a synaptic terminal shown here. And this synaptic terminal sends signal from this neuron, electrical signals from this neuron to the next neuron. Now the electrical signal itself is called an action potential, which is essentially a transient change in the voltage across the membrane. So what really happens is that there is a voltage spike during which the membrane becomes depolarized, which means that the potential across the membrane changes signs and then decays again to the resting potential. This action potential is transmitted along the neuron at very high speeds and without attenuation, so the signal does not drop. And all this is driven by a flow of ions through potassium and sodium channels, which are voltage gated. So let us try to understand this process. So in the image on top, you can see a synapse, and here this is the upstream neuron which is transferring a signal to the dendrite of a downstream neuron. So neurotransmitters are released in these vesicles at the surface of the upstream neuron. These neurotransmitters are released into the space between the two neurons and ultimately bind some neurotransmitter receptors on the dendrite of the downstream neurons. This results in some signaling cascade, which we are not interested in, and opens ligand gated ion channels through which cationic calcium ions start entering the cell. When cations enter the cell, the membrane becomes depolarized. So let's plot this. If you have the membrane potential on the y-axis and time on the x-axis, let's say this is minus 70 millivolts, and this can go up to, let's say, 50 millivolts. So at equilibrium, we have the resting membrane potential. When you have these leak potassium channels which are open, you have a sodium gradient and a potassium gradient across the membrane, and the membrane is negative inside with respect to the outside, and voltage gated sodium channels are closed, and the potential is minus 70 millivolts. When the neurotransmitter binds to the receptor and calcium ions flow in, sodium ions also flow in. When sodium ions flow in right here, the membrane becomes positive on the inside, which means that the potential across the membrane starts to rise. And it reaches a value of about 50 millivolts, during which a lot of sodium ions enter the cell. In an exercise later in class, we will calculate the number of sodium ions that enter the cell. At this point, the sodium ion ligand gated sodium channels start closing, and voltage gated potassium channels start opening. And because the concentration of potassium is high inside and low on the outside, potassium ions start moving outside the cell. This results in a total loss of positive charges in the cell, and then the voltage starts to drop. And eventually it reaches a resting potential like this. We will not talk about this phase right now. I will upload this in a different video later. And now the resting potential is re-established. This transient change in the transmembrane potential is called the action potential. So now we know how an action potential is generated in a local patch of the neuron. But we have not talked about how this action potential actually travels along the axon. So for this, let's look at this image. So here is a neuron. This is the axon hillock where the action potential is first generated. In the resting state, the intracellular fluid is at a negative potential with respect to the extracellular fluid. At this point, at the axon hillock, the action potential is generated because now the cell body has processed the signal it has received from the previous neuron, and there is a net inward flow of positively charged sodium ions resulting from the opening of sodium gated sodium channels. And this results in an accumulation of positive charges in this part, in site A of the axon. Because of diffusion, these cations will diffuse into nearby sites, both to the left and to the right. As they diffuse to the right, the local electrical potential here also becomes depolarized, and this is visible in this image here. As it slowly becomes depolarized, some sodium channels open, which leads to a further flow of sodium channels in this region of the neuron. These ions are flowing inside the axon, and the membrane becomes further depolarized, leading to an explosive flow of sodium inside the cell from these voltage gated sodium channels. So this part of the axon now gets depolarized, and so on and so forth. So it goes from site A to site B to site C to site D. Note that cations will also flow back from site C to site B, not just in this direction, but also in this direction. However, this part of the membrane does not get depolarized for reasons we will not go into. What is interesting to note here is that the electrical signal is passing along this direction, but ions are flowing perpendicular to this direction. So ions are flowing across the membrane in this direction while the electrical signal is passing along this direction. So this is quite different from what happens in an electrical circuit. Hodgkin and Huxley developed a theoretical model where they used the theory from electrical circuits to describe flow of information along neurons, and for which they received the Nobel Prize. They used what is called the cable equation, but we will not go into that in this course, to describe the flow of ions and the flow of electrical signals along neurons.